My name is Simo. I'm a professor of biomedical engineering at the research unit of medical imaging, physics and technology in University of Oulu, Finland. My research group has previously quite extensive experience of working working with the animal or human joint tissue sample, and here in the Miracle project our primary role was preclinical characterization of tissue samples using the mid-infrared spectroscopy. We were also responsible for histological analysis of the disease stage of the tissue samples. Histopathology is the gold standard in medicine for detecting the stage of the several diseases. And here in this project, histopathology was the technique that was used also as a gold standard when we detected the disease stage. During the project, our group has measured hundreds of tissue samples, both from the animal, bovine and human joint tissues. Those samples were primarily articular cartilage, but we have also measured meniscus and ligament samples. With the bovine tissue samples, we artificially simulated the typical osteoarthritic changes within the articular cartilage tissue. We used a mechanical degradation. We scratched the sample surfaces and we also exposed them to impact trauma. And then with the enzymes, we specifically degraded the two primary building components of articular cartilage tissue, namely collagen and proteoglycans. We have also done preliminary spectral analysis for the measured preclinical tissue samples, and aided in the work of identifying the most relevant wavelengths for most sensitive detection of tissue damages, which would then eventually be used in the Miracle device developed in the project. We showed in the project that mid-infrared spectroscopy is capable of detecting specifically both enzymatically or mechanically induced artificial tissue damages from bovine samples. With human samples, we also showed that mid-infrared spectroscopy was able to differentiate the histologically normal samples with the histologically diseased samples. Our results were highly promising, and they give a very good fundamental background for proceeding to further experiments in the project with the newly developed probe, first in the laboratory and eventually during the clinical trials in true arthroscopic setup.